The Indian Institute of Technology Kharagpur, better known as IIT Kharagpur or IIT KGP, was established in the year 1951. Starting off its journey as the first IIT of the country, the institute has earned a reputation of being one of the premier institutes in India. IIT Kharagpur strives to be a hub of knowledge creation that prioritizes the frontier areas of national and global importance that can improve the life of every citizen of the country. As the country started to embark on a path of progress in every field, it became imperative that basic and quality healthcare is made accessible to the people of the country. This required technological advancements in the field of medicine. To meet the challenges posed by medicine in the 21st century, the country needed a new breed of medical professionals who could fuse together medicinal science with technology and bridge the two disciplines. Likewise, engineers with intricate knowledge of the complexities of medical and biological sciences were also sought after. In the year of its golden jubilee in 2001, the School of Medical Science and Technology, SMST, was started at IIT Kharagpur, with the objective to provide a platform of interdisciplinary teaching and research in diverse areas of medical science and technology. School of Medical Science deals with the advancement of medical science for better healthcare delivery in the present scenario and in days to come with newer problems. Medical science deals with one of the most complex systems. Dealing such complexity requires endorsement of the experience of the doctors, the engineering precision and automation with the fundamental inputs from basic research. It also involves the integration of quantitative analytics and computation to make it precise and less vulnerable to decisional ambiguity.
SMST is a very unique department in our IIT Kharagpur system. It started its journey in 2001 when the institute felt its need to start with a department or a school which interfaces the medical world with the world of science and engineering. The flagship program of this particular school, which is known as Masters of Medical Science and Technology, started in that year with a vision to train medical doctors with enough of scientific and engineering skills to make them medical technologists and medical scientists. From very recently, SMST has come up with very unique programs and initiatives to establish link with the broader medical community. One such program is the joint Masters PhD program with the Tata Medical Center, which has started just this year. We have started 3 Masters program or joint Masters PhD programs, and these programs are in the following areas: medical physics, molecular medical microbiology and nuclear medicine. In all these courses, faculty members are jointly from the Tata Medical Center, which is one of the premier cancer institutes in the country and also recognized globally, as well as also from IIT Kharagpur faculty. So we have also started a very niche advanced certification program on clinical oncology, where very senior, where medical doctors from the Tata Medical Center are gaining some research experience through advanced certification program, which they are undertaking under joint supervision of one senior doctor from the Tata Medical Center and one professor from IIT Kharagpur. We have also started the collaborative program or collaborative agenda with the All India Institute of Medical Sciences, New Delhi. The MOU has already been signed and the research agenda, the collaborative research agenda has been exchanged, and we will be soon organizing a joint workshop with AIIMS New Delhi, either at New Delhi or at IIT Kharagpur. Now from the year 2019, IIT Kharagpur will start its medical college cum hospital. It will start with as a super specialized 400 bed hospital, and this hospital will slowly graduate in the form of a medical college.
The School of Medical Science and Technology was founded in early 2000 by IIT Kharagpur under the directorship of Professor Amitabh Ghosh, with a view to fostering cutting-edge research in medical sciences. And for that purpose, the objective was to train a group of doctors and make them familiar with fundamental sciences, engineering sciences, and technology work, so that they can then drive the entire research process. Now, successful culmination of this initiative would obviously be found when the medical students who get admitted into SMST for technology training also have continuous clinical exposure in a specialty clinical environment. And the scientists and technologists who are working hand in hand with the doctors in taking this research forward will also greatly benefit from the actual clinical environment and the actual necessities that arise during patient care, which can be understood only when they are also working and getting exposed to a clinical environment. With that in mind, it took a little bit of time, but with that in mind, finally, IIT Kharagpur took a decision to set up a medical school along with a specialty hospital and a separate research tower where integrated multidisciplinary and interdisciplinary research involving doctors, core scientists and technologists will take place. Very soon, we are going to start with the first phase of this large project. And the first phase involves starting a 400-bed super specialty hospital within IIT Kharagpur. The hospital is built over 400,000 square foot of space. It will have all modern diagnostic and therapeutic facilities, including high-end imaging, high-end critical care, tertiary level operating environment, tertiary level pediatric and neonatal management. Very soon, separate cancer block will also start, which will provide comprehensive end-to-end oncology care. We are expecting the hospital outpatient and diagnostic services to commission very early next year, January, February, and the inpatient services to commission around April, May 2019. With the hospital starting, a very large clinical environment and a large number of doctors participating in the hospital, which will be connected hospital in the sense that there will be a comprehensive hospital management information system which will allow the hospital to connect with the outside world, which will also contribute to research environment very strongly. We have also allocated a space of about 5,000 square foot on the top floor of the hospital for SMST, so that SMST ward is integrated much more closely and progresses in a more seamless manner along with the clinical work of the hospital. So in my opinion, setting up this hospital and starting this hospital today is the true culmination of the objective with which SMST was formed, and this will greatly benefit the students who join us at MMST, the faculty, the PhD scholars and the students who are at the frontier of many innovative and path-breaking researches into technologies which can lead to products or services or solutions.
So my research area is primarily in the domain of microfluidics, which is about fluid flow in small channels. Now what we do with this in the medical world? In medical world, there are several issues or several outstanding problems which we can address through this microfluidics and nanofluidics technology. For example, one can talk of point of care diagnostics. So instead of bringing a patient to an elaborate pathological laboratory, we can bring a small medical device, a handheld device to the patient, take one drop of blood from the fingertip or in some case saliva or urine sample and load that in a device where very quickly we can come up with the results of the diagnostic tests at a very affordable cost. So in our lab we are working on primarily two technologies. One is the paper-based microfluidics, which we have advanced to an extent, and we have been the first group in the world to innovate a new microfluidics technology called as paper and pencil microfluidics. So in addition to that, we are also working on lab on a CD technology, and from point of care we are trying to graduate to extreme point of care diagnostics, that is in extreme rural settings where there is dirt, dust, humidity, extreme heat. Under those conditions you cannot keep your reagents in a refrigerated condition. The entire lab control system will fail. How do you do medical diagnostics in such a challenging condition? So this is one of the areas which my research group is addressing. Another area where the medical world comes into the picture is understanding the biophysics of cancer progression. For example, when cancer is progressing, the cancer cells are metastasizing from one location to other location in a human body. So in that process, it has to proliferate through all sorts of blood vessels, and in the process comes, or in the path comes, microcapillaries. These microcapillaries are like nature-made microchannels. Now, under normal circumstances, a normal cell will not be able to proliferate across that, but a cancer cell can. The question is why. So we are trying to understand various mechanisms of blood flow through small passages, and we are trying to use that understanding to develop a deeper insight on cancer progression, which may perhaps help in better management of cancer. So from cell on a chip, we are trying to graduate on tumor on a chip, and eventually organ on a chip and human body on a chip. And our eventual objective is to replace the animal model experiments with in vitro artificial systems which to some extent can mimic the functionalities of a drug testing platform or a biophysical platform to the extent that is possible to be realized by an engineering system by creating the tumor microenvironment. So these are some of the areas of research. I have some other areas of research which are more fundamentals of microfluidics and nanofluidics, but these are some of the areas which are more application oriented and translational and very much relevant to the main objective and focus of SMST.
I welcome you all at School of Medical Science and Technology, IIT Kharagpur, with a statement: all great revolutions start with a evolutionary impulse. A test run or a trial grounds for the future of humanity. In my research career, I have fascinated towards interdisciplinary research work in the area of MEMS, biomems, sensors, flexible electronics, and solid-state devices for the diverse strategic and medical applications needed for our country. My journey started with a vision to build MEMS-based sensors and to sense the biophysical properties and characteristics of the human body. Our research group has deeply involved in R&D activities including realization of silicon quartz, microelectromechanical systems and microsensors, technology development of the integrated circuit processing, which resulted in successful realization of the several semiconductor devices, integrated circuit, sensors for diverse applications. Presently, giving a prior importance to the biomedical sensing research, major thrust has been given for rapid and cost-effective detection of the diseases in an alternative approach, cultivating the fundamental research for detailed understanding of the chemical, physical, and biological and structural information of the cells in integrated fashion and correlate with the physiological changes in the normal and diseased conditions using miniature MEMS and microfluidic platforms. And thank you all, and we again welcome you to SMST.
My lab is known as the Clinical Biomarkers Research Laboratory. We use high throughput technologies such as proteomics and metabolomics for understanding disease pathogenesis and identifying robust markers to aid clinical decision making. The major areas of emphasis in my lab are respiratory health and women's reproductive health. Some of the valuable contributions we have made is the identification of a very robust panel of markers in serum for detection of endometriosis. Biochemical sensor development based on these findings is underway. Furthermore, therapeutic management of the disease using novel metallocurcumin nanoparticles is also undergoing. We are also in the process of developing a biobank, that is a repository of serum, exhaled breath, bronchoalveolar lavage fluid and lung tissues from patients with various lung diseases. This is to aid cutting edge research, and also we feel that molecular profiling of exhaled VOCs, that is volatile organic compounds, using electronic nose technology will assist in early detection and non-invasive detection of various lung diseases including lung cancer.
I am involved with the research lab and the teaching program on multimodal imaging and analysis for therapy. So why we are interested for multimodal imaging? We consider that any biological system, or you may say the human biology in health and disease, we have to understand from multiple dimensions and in multiple scales. So we need different type of imaging and their analysis to appreciate and correlate, the human domain knowledge with the machine information from the machine that we perform with the microscopy. For microscopic imaging, we perform optical coherence tomography to generate subsurface imaging or subsurface information for the different diseases. It may be oral cancer, it may be different wounds including diabetic wounds, or to study the interaction between cell and the biomaterial matrix that we are developing for the treatment or for the therapy of the different wounds including oral cancers. Our lab has developed different domain expertise, one in the area of the secondary cell and primary cell culture, and to see their relation and the interaction with the different biomaterial matrix including honey-containing matrix with different textures. Our lab also developed, same lab also developed the molecular biology facility and the imaging facility. We have integrated the transbody imaging like ultrasound with the OCT, optical coherence tomography, and stereo zoom microscopy, as well as fluorescent and semi-confocal microscopy. Our researchers are from different background, the engineering background, the pharmacologist, the molecular biologist.
I've been working on the role of actin microtubule crosstalk in the process of epithelial mesenchymal transition, as well as I've been working on wound healing processes, and I've been using light cell imaging technique mostly for this.
We are trying to understand the malignant potential of oral precancerous diseases based on medical heuristics, histopathological and molecular information, and optical coherence tomography.
I am exploring the involvement of reactive oxygen species in the conversion of pre-malignant oral squamous cells to malignant carcinoma. I am working on epithelial mesenchymal transition on oral cancer and cancer. Epithelial mesenchymal transition is a very important process for cancer metastasis. Here initially we have developed a in vitro morphological model. Then we are trying to translate the knowledge in histopathology.
Modalities. One of the imaging modalities that I am working on is optical coherence tomography. The role of optical coherence tomography is to get cross sections of the tissue optically without doing any physical biopsy. What we can see is underlying structures. As you can see here, this is an image of skin and from the palm, and we can see different layers very clearly. And in terms of cancer, in terms of regenerative medicine, we can see how the wound heals, we can see how the tumor is progressing, and this way we are trying to develop. This, for instance, is the image of a carotid artery along with the jugular vein seen in color Doppler imaging, and this shows the blood flow in the blood vessels. So things like that is incorporated in the education.
We are trying to develop an index which will help doctors to assess the malignant potentiality of oral cancers precisely. And we are incorporating histopathological and immunohistochemical features to achieve this.
I am a doctoral research scholar of Professor Diploma Chatterjee, and we are using here optical coherence tomography machine for non-invasive real-time imaging purpose and to coordinate the images with the gold standard of histopathological images.
And I am Neha Sharma. I am working in the spectroscopic and imaging techniques in the analysis of biomarkers of oral cancer. And this is one of the picture of dried saliva, where we find that if there is a difference between the layers of the crystallization happening in saliva.
I am a doctor who is pursuing a master's program here for research, and I am working with research scholars here for developing methods on early and non-invasive diagnosis of oral precancer and cancer by characterization of molecular signatures derived from chemometric analysis of data which are obtained from saliva samples by spectroscopic and imaging methods.
Professor Santanu Dhara. I joined this institute back in 2007. He was in ceramic fabrication from IIT Kharagpur at Material Science. Mostly we were working on colloidal processing of ceramics, which is basically a powder metallurgy route, and I was majorly working on different type of product developments based on gel casting, and there I developed a process called protein coagulation casting, and where we have used the egg white, and simply just using egg white we are able to make components which is of 99.9% density as well as more than 96% porosity, which has diverse application from space to defense to structural applications. Once I joined the School of Medical Science and Technology back in 2007, so we formed a group which is a highly interdisciplinary research team where people from mechanical engineering, from bioscience, microbiology, tissue engineering, materials as well as physics, and many other departments people have joined here, including pharmacy, for the product development. So my group major focus is on product development, while mostly we work on products for rehabilitation, products for better healthcare in a cost-effective way. So here major motivation is to develop the products by developing the platform technologies. So really our group is working on diverse waste material for value-added healthcare products. As you know that most of the products which are available in the market, the raw materials we usually purchase from foreign advanced countries like Sigma, Rich, Goodfellow and so on and so forth. So with that, because of the starting materials is very expensive, when we make the product becomes very expensive. To make the cost-effective products, we actually rely on biological waste materials, we recycle them and finally develop the technology for healthcare products. So in this domain, we work on bone tissue engineering, skin tissue engineering, we make the skin grafts, we also make the nano carbon dots, we make carbon dots for live cell imaging, we also make it for raw scavenging and for other healthcare applications including hand wash. And also we are extensively working on 3D printing of live cells and the materials for organ printing. So we are basically working on 3D printing of tissues and 3D printing of titanium as well as 3D printing of polymer implants. Specifically, we have extracted collagen from fish waste material. Using the fish waste collagen, we have developed series of products like collagen flake and collagen sponge as well as the dressing materials for diabetic, burn wound as well as different other healthcare applications. We make the transient tissue engineering scaffolds where we seed the cells, we isolate the cells from placenta, then seed on the materials, and the cell seeded materials we graft at the site for tissue integration. Similarly, we have used the chitosan, which is actually a crab cell waste, and using this we developed a spinning technique by which we develop the fibers for healthcare like nutrition. This is a ceramic fibers. We have developed a technique for spinning the ceramics, and we developed the ceramic fibers, and these ceramic fibers and the resin together we made the composite, and using this composite we developed a customized mandible through the reverse engineering approach, where we have used the CT image, and using the CT image we extracted the 3D model of the desired tissues, and then based on that extracted image of the organ we developed the composite mandible, we developed the ceramic mandible, as well as we developed 3D printed spinal case, as well as 3D printed maxillofacial products. And we developed a process called green machining of ceramics, by which we have made dental crown, which is a customized crown, and the uniqueness is that we developed a tool by which you can do the machining of ceramics, and the ceramics we developed which is highly machinable. And also we developed a series of products based on titanium, like dense titanium, porous titanium. So this is a dense titanium, this is a porous titanium, and this is like dense and porous sandwich titanium. As you can see here, this is a Zimmer implant which is actually failed from the patient. So they make it porous so the tissue can grow into the pores. So here we have developed very cost effective way, a simple method for making a porous titanium, and we have made it bioactive. So in this porous titanium, tissue can grow into it and it can be having the integration with the bone, and we call them osteoincorporation. Okay, with that I can end. Thank you.
Medical instrumentation lab, we are working on developing assistive devices for post-stroke rehabilitation. We try to address the condition known as hemiplegia in a patient who suffer from foot drop cases. So in foot drop case, we employ the external stimulation to the peripheral nerve and the tibialis anterior muscle to bring in the dorsiflexion in affected foot. And in this way, the patient will be able to walk normally. We have placed an external pacemaker-like device, a stimulator, which will stimulate the peroneal nerve and the tibialis anterior muscle in such patients to bring in dorsiflexion. In addition to the post-stroke, the other assistive device we are into is on developing a exoskeleton device for post-stroke rehabilitation purpose, where the patients will be, trained by using this external exoskeleton device. So in this device, we need not depend mainly on the physiotherapists who are, sparingly available, so that we can make use of this exoskeleton device for post-stroke rehabilitation. So in addition to this, we are also into developing other non-invasive devices like the impedance cardiography for estimating the cardiovascular parameters. Currently, we are having a project to develop this exoskeletal device in collaboration with Medical College, where the clinical trials will be carried out to assess the use of this exoskeletal device in post-stroke rehabilitation.
My lab is working in different aspects of cancer. So in cancer, actually that is a global problem. So all the chemotherapy drugs people are using, they are toxic and there are a lot of side effects. So they are killing also the normal cells. So that's why actually our lab is aiming to find out some drugs from the natural source which has the least toxicity. So we have published several papers, thymoquinone, curcumin and other natural products in different reputed journals. And also we are working actually the targeted drug delivery. With chemistry department we are working, and there are different type of, we are making different type of gold, silver and different type of nanoparticle and conjugated with the drugs and the targeted drug delivery, and that way we can reduce the toxicity and specific drug delivery. Our lab is working on the mechanism of chemoresistance. So different people, and we have published reputed paper in Oncology, Cancer Letters and so many good reputed journals. And what is the mechanism of cancer chemoresistance, and that's why actually, and some of my students they are working on autophagy and other aspects of cancer. One group is working on glioma, and another group is working on breast, another group they are working also on cervical cancer.
Hi, I'm Sangeeta Bhattacharya. I am a professor here in the School of Medical Science and Technology, and good luck with Research Scholars Day. Some key members of our group, we are working on different issues related to global health, and here behind the computer is Dr. Dila Khan. Dr. Khan is a virologist, and right behind you over there is Tushar Mundley. Tushar is a PhD scholar, and right here is Ranjan. Ranjan is a medical social worker, and we are working on lots of different things this morning. Dr. Khan is working on developing a framework to make sure that everybody in the department is immunized to hepatitis B. Tushar is working on a clinical decision support system to improve dosing in children with acute and relapsing leukemia. And Ranjan is working on our hepatitis B project, which is the one that's all of the data collating and collecting for our entire department, and we wish you all the very best. Thank you.
I'm heading the Regenerative Medicine Lab at the School of Medical Science and Technology. Anything that involves regeneration is where my interest lies. One of the areas that I'm very much interested is in the beta hemoglobinopathies, where thalassemia is my area of interest, in which we are trying to look into how microRNAs and cell signaling pathways can be utilized to improve the fetal hemoglobin levels. The other area that I am working is in the field of osteoporosis. So basically what we are trying to see is osseointegration in these patients in case there is a fracture. So there is obviously a need for an implant, but many of the times these patients have a problem in the bone grinding property of these implants. So if we can actually coat these implants with some specific drugs which have been known to improve osseointegration, then that's definitely going to be helpful. I'm also trying to see if we can develop some easy user-friendly tools which can be provided to grassroots level workers that can help them collect data. So in this area we are trying to work in maternal and child health area, where we are trying to develop a simple decision support tool for these healthcare workers, the grassroots level workers, but they can use this tool to try and collect data for pregnant women for whom these grassroots level workers are the first line of contact.
Basically, I'm trained in immunology, and I envision a research path for myself in basic immunology as well as development of novel immunotherapeutic strategies and reagents. The first project that we are working on is the development of an immunotherapeutic reagent, which is basically an artificial antigen presenting system that will help reprogram exhausted or inactivated T cells, which are going to be used as a cancer immunotherapeutic strategy. The next thing is that what we are working on is looking at studying the mechanism of immune modulation by different important biomolecules, like especially vitamin D, in inflammatory diseases. Right now we are concentrating on autoimmune hepatitis. Thirdly, what we are working on is the enumeration of the role of an Ig superfamily member, which is an important T cell inhibitory molecule. What we are planning to do is to analyze or study the role of this molecule in inflammatory disease like ulcerative colitis, and also to help find its binding partner using different techniques like immunological as well as biochemical techniques.
I'm working on heart failure and atherosclerosis. Particularly, I'm looking at one immune cell which is called natural killer T cell in the development of atherosclerosis. For this, I'm using mouse and cell culture. Second, if you see the burden of disease in India, atherosclerosis or cholesterol-related problems have gone up. In that direction, we have a lot of drugs, and the newest drug is called PCSK9 inhibitor. I and my team here in IIT Kharagpur are working on PCSK9 inhibitor drugs and how these drugs would modify cardiac remodeling after ischemic stress. And I hope that with these two, the development of atherosclerotic plaque and natural killer T cells and PCSK9 inhibition in cardiac remodeling, we hope that we will make some better understanding, we will get better understanding of how these diseases are manifested and probably can be prevented in the future. Thank you.
Medical decision making suffers from ambiguity and imprecision. Towards addressing this problem of decision making, medical experts contact with mathematicians and statisticians, because mathematics and statistics have a lot of approaches to resolve those issues. However, the fundamental aspect is to model uncertainty, because uncertainty is an unavoidable phenomena in medical decision making problem. And in this respect, among different approaches, fuzzy logic plays an important role as it has certain interactive interfaces with human. Fuzzy logic has powerful capability to model human perception, thinking, reasoning, etc. Now present days we are dealing with analyzing the malignant potentiality index, and here you are using fuzzy cognitive map approaches, fuzzy information fusion strategies, level identification. Our main aim is to make a robust diagnostical support system.
Achieving quality knowledge continuum where nothing is valueless and considering failure as an important step towards future success, SMST is earnestly trying to evolve the ambience of collective learning, with a vision to develop appropriate state-of-art technologies in healthcare and a deeper multidimensional understanding of medical mysteries.